Hi, my name is Pam Guerrerio, and I am Chief of the Laboratory of Allergic Diseases at the NIH. For several years now, my lab has been interested in studying the role of TGF-beta in the immune system and how alterations in TGF-beta signaling might contribute to immune-related diseases in humans. And so I'm very honored to have this opportunity to tell you about our plans to study the immune phenotype of patients with Myhre syndrome and also share with you what we have learned so far. So I'm sure many of you are already familiar with the typical clinical manifestations of Myhre syndrome with involvement of the cardiovascular, respiratory, GI, skin, craniofacial, neuropsychiatric, and skeletal systems. But what's missing from this list is the immune system. So TGF-beta has long been known to play a critical role in both the development and the function of the immune system. But whether patients with Myhre syndrome have a specific immunophenotype has not been well studied. In the literature, I could only find one report that reviewed 32 patients who had pathogenic variants in the SMAD4 gene, including 30 patients with Myhre syndrome. Of these 32 patients, over half of them, 53%, reported recurrent ear and respiratory infections. Two of the patients had a formal diagnosis of humoral immune deficiency or antibody deficiency, although antibody levels were not systematically evaluated in the entire cohort of patients. And interestingly, in two of the patients, the recurrent infections were complicated by choanal atresia. And once this was repaired surgically, the frequency of their infections did decrease. This suggests then and leads to this question of whether the increase in infections reported by Myhre patients is driven by an underlying immune deficiency or whether it might be a product of their propensity for fibrosis and altered craniofacial anatomy or both. So Myhre syndrome is an autosomal dominant disease caused by pathogenic variants in the SMAD4 gene. The SMAD proteins constitute a family of different proteins that work together to regulate gene expression. One class of these proteins are the receptor-regulated SMADs, which include SMADs 1, 2, 3, 5, and 8. SMAD 6 and 7 are the inhibitory SMADs, and then SMAD 4 is a co-mediator. So activation of the TGF-beta and BMP receptors by their ligands leads to phosphorylation of the regulatory SMADs. They then form a complex with SMAD4, and these complexes translocate to the nucleus where they regulate transcription of a whole wide range of different genes. The SMAD4 protein is composed of two domains, the MAD homology or MH domains 1 and 3. The MH1 domain is required primarily for binding of SMAD4 to DNA, and the MH2 domain is required for SMAD4 to regulate gene transcription. The variants that cause Myhre syndrome have all been largely in two specific residues in the MH2 domain. So the effect of these mutations on the stability and function of SMAD4 is really not completely understood, but it's thought to increase the stability of SMAD4 by reducing ubiquitination of the protein, which normally would target it for destruction. And indeed, in fibroblast cells from patients with Myhre, it has been observed that they have increased levels of phosphorylated SMAD2/3, as well as phosphosmads 1, 5, and 8, which are downstream signaling molecules in the TGF-beta and BMP pathways, respectively. However, interestingly, this was actually associated with lower expression of genes that are typically regulated by TGF-beta, suggesting that these mutations in SMAD4 somehow also inhibit the ability of the protein to properly regulate gene expression. Well, what does this all mean for the immune system? So as I mentioned earlier, TGF-beta plays a very pervasive role in regulating immune responses by influencing the development of and function of cells both in the innate immune system, including mast cells, monocytes and macrophage, eosinophils, basophils, neutrophils, and NK cells, but also has a very important role in the adaptive immune system as well, including the function of both B cells and T lymphocytes. So in fact, TGF-beta has been shown to be one of the key cytokines that regulates the differentiation of naive T cells, where it promotes the development of T helper 17, Th9, and T regulatory cells. while inhibiting the development of Th1 and Th2 effector T cells. So each of these different types of effector T cells have a unique job. The Th17 cells are essential for protecting the host against fungal and extracellular pathogens, but they can also contribute to autoimmunity. Th9 cells have been more recently described, and they have a diverse role that's ever growing. They are very important in our protection against extracellular parasites, but they also seem to have a role in anti-tumor responses as well. People with autoimmune and allergic disease have been shown to have an increase in the frequency of Th9 cells in their blood. T regulatory cells, on the other hand, have a role in promoting immune tolerance, both to self antigens as well as to innocuous antigens, such as those found in food and commensal bacteria that normally live in our skin and gut. So, on the other hand, TGF beta inhibits the development of Th1 cells, and these help protect us against intracellular pathogens. But again, like Th17 cells, they can also contribute to autoimmune responses. And then TGF beta inhibits Th2 cells as well. And these are really the key cell types that have been implicated in allergic disease, but they evolved to help us fight parasitic infections. So in the big picture then, TGF-beta really has both pro and anti-inflammatory properties. On the one hand, it promotes tolerance by driving the differentiation of Treg cells and inhibiting T cell proliferation, activation, and differentiation, at least of Th1 and Th2 effector T cells. But on the other hand, it has a pro-inflammatory role in promoting the differentiation of Th17 and Th9 cells. So most of the information about how a disruption in TGF-beta signaling might affect the immune system has really come from mouse models. There are various lines available of mice that are deficient in TGF-beta signaling as a result of reduced signaling primarily through the TGF-beta receptor. But the phenotype of all these mice is really quite similar. In general, what we see is the mice develop this profound inflammation that affects multiple organ systems. And this is associated with very high levels of autoantibodies and immune complex deposition that generally results in death of the mice at a very young age. A lot less is known about how defective TGF-beta signaling would affect immune development in humans. But for the last several years, we have been studying the immune phenotype of patients who have an autosomal dominant connective tissue disease called Loeys-Dietz syndrome. LDS is caused by mutations in genes encoding the receptor for TGF-beta, either the first or second subunit. LDS was initially defined as a triad of hypertelorism or widely spaced eyes, a cleft palate or bifid uvula, and a propensity to develop aneurysms throughout the arterial tree, which is the primary cause of early morbidity and mortality in these patients. Most of the mutations that cause LDS are missense mutations that occur in highly conserved residues in the kinase domain of the protein, with the majority, about 3/4 of the mutations identified, being present in TGF-beta receptor 2. The effect that these LDS mutations have on signaling from the receptor is still something that's being actively investigated. But at least in immune cells, we have found that these mutations seem to result in reduced TGF-beta signaling. So here you can see that we found significantly reduced levels of phosphorylated SMAD2/3 in naive T cells from LDS patients, shown here in blue, compared to age-matched healthy controls following stimulation with TGF-beta, suggesting this results in a loss of TGF, or at least impairment of TGF-beta signaling. So given what I had told you earlier about the consequences of reduced TGF-beta signaling in mice, we expected to find a higher prevalence of autoimmune disease in LDS patients, given that TGF-beta signaling seems to be impaired. But what we actually found is that these patients have a very high prevalence of essentially all forms of allergic disease. So in our original cohort of about 80 LDS patients, we found that just under 40% had been diagnosed with eczema, nearly half with seasonal allergies, 45% with asthma, just under a third with food allergy, and nearly 10% with eosinophilic esophagitis. And you can see the rates of all these conditions were significantly higher than what we generally see in the general population. So, consistent with their allergic tendencies, we found a higher number of circulating plasmablasts as well as B cells secreting both IgG and IgE antibodies in the blood of LDS patients compared to age-matched controls. And this was supported by higher levels of both total serum IgG and IgE in patients with LDS compared to controls. So we next wanted to try and understand how this decrease in TGF-beta signaling might be promoting greater antibody production in LDS patients. And over the last several years, it's become quite clear that T follicular helper cells, or TFH cells, play a key role in dictating both the nature and the magnitude of humoral immune responses. TFH cell development occurs in secondary lymphoid tissues where naive T cells encounter antigen-presenting dendritic cells in the T cell follicle. These cells upregulate a transcription factor called BCL6, which primarily drives TFH development, as well as CXCR5, which is a chemokine receptor that promotes the migration of these pre-TFH cells to the T B cell border. Here, additional signals from the B cells further promote commitment of these cells to a TFH cell program, allowing for the formation of germinal centers within the B cell follicles, where B cells undergo affinity maturation and class switch recombination to generate high affinity antibodies. So there are thought to be different subsets of TFH cells, which in humans can be distinguished by the expression of chemokine receptors, including CX3CR1, CCR4, and CCR6. Each of these different subsets secretes a particular set of cytokines reminiscent of the corresponding T effector cell subsets, and these cytokines can promote class switching in a particular direction. For example, TFH2 cells produce IL-4, which promotes class switching to IgE. In addition, the proliferation of TFH cells in germinal cell activity is opposed by a class of cells called T follicular regulatory cells, which share features both of TFH cells and Treg cells. Now, among the cytokines that have been found to regulate TFH development in both mouse and humans is TGF-beta. And so we hypothesized that alterations in TFH development might be contributing to the enhanced antibody responses that we observed in LDS. And indeed, we found that the total number of TFH cells was increased in the blood of LDS patients compared to age-matched controls. Furthermore, by looking at that pattern of chemokine receptor expression that allowed us to discriminate the different subsets of TFH cells, we found that the increase in TFH cells in LDS patients was specifically in the subset that promotes Th2 immunity and IgE production. There was no difference in the TFH1 cells, and TFH17 cells were actually decreased. Well, what does this all mean for Myhre syndrome? Well, we have an ongoing study now. It's still very early in our research, but we're hoping to try and do the same type of detailed immunophenotyping in patients with Myhre that we had done in LDS to understand exactly how these mutations in SMAD4 are affecting the development of immune cells. So far, we have had the opportunity to look at antibody levels in 4 patients with genetically confirmed Myhre, and they're represented here in these graphs. Each dot is a different patient. And what you can see is we saw the exact opposite of what we had seen in LDS. In general, the Myhre patients have very low IgG and IgA antibody levels below the 95% confidence interval for age, which is what these lines here are representing. They also had very low IgE levels, which again is opposite to what we had seen in LDS. We also had the opportunity to look at the ability of Myhre patients to respond to vaccines in these few patients. And 4 out of the 4 patients did have protective titers to tetanus. We had data on 3 patients with diphtheria, and all 3 of those patients did show protection against diphtheria, but only 1 out of 3 had protective titers to varicella. Now, this is a caveat to this is we are not completely clear whether these patients that did not have a proper response to varicella, whether they had actually received the vaccine. Now, interestingly and very importantly, all four of the patients did report recurrent ear and sinus infections, and all of them had at least one episode of pneumonia. So the question now is whether this decrease in antibody levels that we're seeing is somehow contributing to this infection, or are these two observations true, and unrelated? So, based on this limited data, then, the concern then is whether Myhre patients are at higher risk for a condition known as common variable immunodeficiency, or CVID, which can predispose patients to recurrent infections primarily involving the ears, sinuses, and lungs. CVID is an immunodeficiency disorder that is specifically characterized by defective antibody production. The diagnosis of CVID requires that the patient have reduced IgG levels in addition to low levels of either IgA and or IgM. But a second key piece to this diagnosis is that the patients must show poor or absent responses to vaccines. So from the few patients that we have been able to evaluate so far, we do see that at least 3 of the 4 patients met this criteria for having low IgG levels along with low IgA. But what we're not yet clear is whether they are not able to mount an appropriate response to vaccines. And that's where we really need to do more research to understand if that is also the case. And so I would like to end my talk with a thank you and a request. So first, I want to say thank you to the patients and the researchers who have been so gracious with their time and talent to support our research study to better understand how the immune system is affected in Myhre syndrome. We absolutely could not do this work without you. And second, I want to ask you to consider helping us continue our research studies at the NIH to help us learn whether patients with Myhre syndrome are indeed at greater risk for infections and how this disease might affect their ability to respond to vaccines, as well as their risk of autoimmune and allergic diseases. So for this part of our study, we can actually collect much of this information through a telehealth visit. But for those patients who might be able and willing to come to visit us in Bethesda in Maryland at the NIH, we would like to do several blood studies to try and better understand exactly how these variants in SMAD4 are affecting the immune system at the cellular level. So looking at how it affects the function and development of both T and B cells, and specifically antibody production. I also wanted to take this opportunity to tell you about another but related study that we've recently started working on with our colleagues in NIAID. And this study is specifically designed to assess the immune response to the COVID-19 vaccines in patients who have various genetic disorders that affect the immune system, including Myhre syndrome. So patients can participate in this study either by coming to the NIH for an in-person visit and blood draw, or we can actually send a finger stick kit to their home and the patients can mail their samples back to us. So if you or your patients are interested in either of these studies, please contact me. We would love to have you participate and help us learn more about the immune findings of Myhre and hopefully identify ways that we can improve the care of this patient population. So finally, I just want to recognize the members of my research group shown here who performed the work I showed today. I also want to recognize our collaborators at Hopkins, but especially Doctors Angela Lin and Mark Lindsay at Mass General, who have been such wonderful collaborators, as well as Dr. Koja at Northwestern University, who have been instrumental in helping us with our studies. So thank you very much. I'm looking forward to hearing your questions.